Okay, so I want to continue our video on this review paper, which is iron regulatory protein, iron responsive element signaling pathway in human neurodegenerative diseases. So, basically, to do a quick recap on the previous episode, we went over what these iron responsive proteins or these iron regulatory proteins are and how they influence the translation of the mRNAs that have these iron responsive elements on them, whether they upregulate them or downregulate them, depending on where they are located at on the mRNA, and also whether or not, if you have sufficient iron in that particular area, that will determine whether or not these proteins are going to be on or off. So, with that, let me just get a drink and we can continue. So, these iron regulatory proteins can act as either a translational enhancer or translational inhibitor. In iron deficient cells, the interaction between the iron regulatory proteins and the uh, IREs, the iron regulatory elements, or the iron shoot, let me double check that. Iron responsive elements. Sorry, it's sometimes you will see both iron responsive proteins and iron regulatory proteins, and so basically it's all cramming into the same acronym for me. Anyways, the iron responsive element motifs in the 5' untranslated region of target mRNA can abrogate translation through interruption of important interactions between the mRNA and the ribosome for the initiation of translation. However, in iron-replete cells, iron can bind with the iron-responsive proteins or iron regulatory proteins to induce a conformational change, which promotes the dissociation or the removal of these iron regulatory proteins from the target mRNA. This is leading to a facilitation of the translation of the target mRNA. So this is what's happening in this first situation. So normally the iron regulatory protein, if it's binding to the iron response element, in the 5' untranslated region, what it's doing is it's getting in the way from the ribosome and the other machinery that is required to have this mRNA be translated into protein. So normally translation is going on and you're producing your protein, but this iron regulatory protein is binding to the iron responsive element, which is preventing the ribosome and all the promoter machinery from getting on there properly and allowing that translation process to happen. But if there is iron, or if you're in an iron replete state, this causes the dissociation of the iron response protein or iron regulatory protein, which then frees up the 5' end, which allows for the ribosome and the other machinery, the promoter related machinery, to do its function. So then you get a protein in this particular case. So in contrast, some mRNA transcripts contain one or more iron response element motifs in their 3' untranslated region. So this is the 3' region over here, and this is what you can see in D, E, and F. So in iron deficient cells, the binding of IRP and IRE to an IRE at the 3' untranslated region of transcripts can protect target mRNA against endonuclease cleavage. So endonuclease, what are those? So endonucleases are enzymes that will break up or hydrolytically break up polynucleotides. And it's an endonucleotase, which means that it happens at the inside of nucleotides. So these endonucleases are enzymes that cleave the phosphodiester bond. So here we have an example of, this is probably, it's either DNA or this is DNA. And here you have your phosphate groups. And so these are called phosphodiester bonds because this is one ester bond or phosphoester bond, and this is another phosphoester bond. So this is these bonds are between the alcohols that were originally on the ribose and the phosphate group, which sort of is like an ester group in some way. And if you're not familiar, an ester group is a carboxylic acid that has reacted with an alcohol to create a normal ester group, but this is a phosphate group instead of a carbon group, so it's a phosphoester, and in this case it's a phosphodiester bond. Anyways, this phosphodiester bond can be attacked by endonucleases, which then split it up, and this is one of the ways that your body or life in general will regulate the mRNA and regulate DNA as well, but regulate mRNA to prevent, what would the word be? If you wanted to stop the production of a protein, well, one of the ways to stop the production of a protein is to break up the mRNA that is encoding for that protein and preventing that from building up. So in normal circumstances, you have these iron regulatory proteins that, if they have a lot of this secondary structure in them, this can help signal to the endonucleases, hey, we need to destroy this. So this is functioning like double-stranded RNA, and double-stranded RNA is very often attacked by the body or by the cell's machinery because it looks similar to a virus. Some viruses have double-stranded RNA as their genetic code, and so that would be one of the ways that your body is able to recognize foreign material. And so, how should I word that? So very frequently during a virus's life cycle, if it's an RNA virus, there will often be periods of time in that duplication cycle where you will have double-stranded RNA, where you'll have one strand of RNA that might have come from the virus originally, but then in the duplication process, the polymerase process, you end up creating a double-stranded RNA, and those double-stranded RNAs are some of the, what would the word be? Signals that cells can look for to recognize a pathogen. Now, granted, we're just saying that it's to recognize a pathogen, but it doesn't necessarily need to be a pathogen. Some mRNA normally in your body will have these regulatory elements which can be double-stranded in certain locations, and that might also be able to what would the word be? Imitate and latch onto that system so that they can be degraded by the same enzymes that can degrade the viruses. So I think it's in biology, and I don't like this frequently, but we have to do it. In biology, we have to sort of ascribe a goal to an enzyme, or we need to ascribe what is the psychology of these inanimate objects, right? Because these are all genes, proteins, chemicals. And so these genes, proteins, and chemicals, they're just doing their thing, not necessarily with a goal in mind. They're just doing something random almost, and the things that are good for the cell survive and then get passed on throughout time. So we as researchers and what would the word be? We as people who are trying to figure out why something has stayed in the population, very frequently we ascribe a characteristic or a goal, an end goal for that particular gene, protein, or chemical. And so the problem with that is that once you say this is the goal, or this is the reason why this gene still exists, then people think, that is the reason why this gene or this particular enzyme exists in the first place. But granted, saying you have one function or saying this is the function is extremely disingenuous. That isn't always the case. Sometimes all these different chemicals and genes and proteins, they're doing many different, probably an untold number of different functions. We just try to find out what are the most important ones and then write articles about it. But really, they could be doing a plethora of different things, and we just, if we had the ability, we could discover as many of them as possible. But really, as I said, it's a noisy field. It's really hard to separate all of the different functions of a singular protein from each other. So I'm rambling a little bit, but just know that this process of finding double-stranded RNA is conserved in a lot of cells, and this conservation is being mediated, or this conservation seems to have multiple benefits in different areas. And so, granted, I shouldn't say this explicitly because I don't know if the exact enzymes are working on these, if the same exact endonucleases are working on these particular mRNAs, or if there's bleed through with the viral mRNAs, double-stranded mRNA. But I would say there probably is at least a lot of good conservation between those enzymes if they are different. Excuse me. Anyways, let's continue. Sorry, I'm rambling. So therefore, the interaction between the iron regulatory proteins and the 3' untranslated region IREs can extend the half-life of, and half-life, think of this as like the half-life of radioisotopes. So over time, after an mRNA gets synthesized, it'll have some time before it just randomly gets destroyed. And if you have a lot of these iron regulatory elements, the half-life is shorter. So it is easier for them to be broken up. But if you have these iron regulatory proteins on them, the half-life is longer. So it takes more time for this mRNA to be destroyed. Anyways, therefore the interaction between the IRPs and the 3' untranslated region IRE can extend or increase the half-life of transcripts and promote translation of target mRNA. However, in iron replete cells, the dissociation of IRP from the iron response element at the 3' untranslated region renders the target transcript susceptible to endonuclease attack and degradation, leading to downregulation of translation of transcripts. And the transcripts are the transcripts are the part of the mRNA that will encode for the protein. So here it's shown in red. Anyways, both transcripts of ferritin and ferroportin have IREs in the 5' untranslated region, so that under iron deficiency conditions, the translation of ferritin and ferroportin can be inhibited by the iron regulatory proteins, and that makes sense. If you do not have that much iron floating around in the cell, you don't need ferritin to store it, right? So if you are deficient, or not necessarily deficient, but if you don't have a lot of free labile iron floating around, then you don't need to produce more ferritin to store that extra iron because it doesn't exist. And likewise with the ferroportin, if you do not have a lot of iron in the cell, you don't need to have these ferroportins to basically pump out the iron from the cell into the extracellular matrix. Anyways, both of these can be inhibited by the iron regulatory proteins. Anyways, the decrease of expression of ferritin and ferroportin can reduce unnecessary iron binding by ferritin and iron export by ferroportin, leading to an increased level of free iron available for the cell usage. However, both transferritin receptor and the DMT1, which is the divalent metal transporter 1, mRNA, these have the 3' untranslated region iron response elements, which can bind to the IRPs under iron deficiency. This is leading to the stabilization of transcripts and subsequent increase synthesis of transferrin receptor and divalent metal transporter 1. And this promotes iron absorption into cells. So remember, transferrin is the protein that helps transfer iron throughout the body. And so the transferrin that's floating around outside of the cells has iron, usually it has iron bound to it. Now what happens is this transferrin will bind to this transferrin receptor, and then by binding to the transferrin receptor, you have that endocytosis process happen, where you basically suck up and create a bubble that has the receptor and the transferrin stuck on it. And this, if I'm remembering correctly, this DMT1 is also going to be present on the cell surfaces. And then after that endocytotic event, this you can undergo the process of liberating the iron from the transferrin and then pumping it into the cytosol. So that is, so that is that. And this promotes iron absorption into cells. So if you do not have enough iron, you are now stabilizing these proteins, which is encouraging iron sequestering into the cell. So in contrast, under a situation of iron accumulation, the increased iron level can disturb the IRP-IRE interaction to promote the translation of the ferritin and ferroportin transcripts, as well as destabilize the transferrin receptor and DMT1 mRNA. And I believe transferrin itself is also a part of that. So let me look this up real quick. Iron response element. let us look up the iron. Okay, so there used to be, I shouldn't say used to be, I had another video on it, and I'll probably have to go to that as well. There was a really nice diagram that I found that showed what proteins are up and down regulated in the presence of iron. So let's say iron regulation. So, and you know, if we can't find it, it's all right. For some reason, my internet's not working well, so we'll just continue recording. So, just know, I believe transferrin is also regulated in response to the presence or absence of iron. So, therefore, under iron accumulation, the iron absorption will be inhibited, while iron storage and export can be enhanced. Pathological factors include inducing a disturbance of the IRP-IRE signaling pathway will impair iron homeostasis, which can contribute to the onset and development of human disorders. So if this process gets dysregulated, you can get some pathology resulting from that. However, one recent study reported a novel mechanism for the iron-induced modulation of translation of target mRNA. They demonstrated that eukaryotic initiation factor 4F, or EIF4F, can, and I'm going to make note of that, this can specifically bind to the iron response element at the 5' untranslated region of target mRNA with high affinity, which is vital for translation initiation. So, and this is on the 5' end. So the presence of this will really increase translation somehow. However, iron can also directly bind to the iron response element of target mRNA, leading to a conformational alteration of mRNA. So that's also interesting that iron in of itself can bind to these iron response elements. And you know, that kind of makes sense because iron is positively charged and the RNA is negatively charged. So there could be some binding that just happens through random electrostatic interactions. But what I would presume is that these iron responsive elements have somehow are able to imitate or facilitate iron binding directly to it. Anyways, the conformational changes in mRNA induced by the binding of iron will facilitate the interaction between the EIF4F and the IRE RNA. Interesting, and which can outcompete binding with the iron response proteins and the iron response elements. So interesting. So if you have iron and this EIF4F, that could outcompete the binding with these iron response proteins. So that might be an interesting field to look in later. Therefore, accumulated iron can contribute to upregulated translation of target mRNA with 5' UTR IREs. So this is actually very interesting. So normally, if you have plenty of iron in the system, then this will kick off these these iron regulatory proteins, which if your iron response elements are in the 5' end, oh wait, the 5' end over here. So no, that still makes exact sense. So here we have it in the 5' end, you kick it off, you have normal translation. Well, it appears as though there could be a secondary mechanism at increasing translation where you have the, wait, what's it called? The the EIF4Fs. So if this EIF4F protein can actually also bind to the IRE, and in the presence of iron, you can get a lot of it binding together. But rather than suppressing translation, you're actually initiating or promoting translation. So yeah, that's very interesting. So recent evidence suggests that the IRP-IRE signaling pathway may play other physiological roles beyond iron homeostasis. Functional IREs are identified in both transcripts of CDC14A, and this is cell division cycle 14 homolog A, which is just another protein, and hypoxia inducible factor 2A, or HIF2A. HIF2A. Anyways, the translation of CDC14A, which is a phosphatase involved in cell cycle. This can be modulated by the iron regulatory protein iron responsive element signaling. So this CDC14A, which is associated with the cellular cycle, so mitosis, right? It's one of the proteins that facilitates in a particular stage of the cell cycle. This is regulated by this IRP-IRE cell cycle. So, and also the hypoxia-inducible factor, which I think this HIF2A is actually extremely important, especially in the context of coronavirus, but I've done a previous video on that, and you can go watch that if you want. So, the iron depletion can lead to growth inhibition at the G1S transition. So the G1S transition, just know that this is a part of the cell cycle. So this is the growth, or one of the growth stages, and this is, how do I phrase that? If I remember all this correctly, this growth stage is when the cell just gets on bigger and produces proteins, but right here, at the S stage, this is where you start getting ready for mitosis. You start duplicating the DNA and getting everything in place, and then you can go into the G2 phase, which basically just more growth after you've done all of your tidying work, and then you undergo mitosis, which is basically you do mitosis and then you're back in the G1 phase. Now let me double check this. So the G1 transition, so in the mid-late G1 phase, cyclin D bound to this activates the expression of the S-phase cyclin CDK components. However, the cell does not want S-phase cyclins to become active in G1. Therefore, you have this inhibitor. Let's just look at what the S-phase is. So, synthesis phase. Okay, so this is a phase of the cell cycle in which DNA is replicated occurring between the G1 phase and the G2 phase. Since accurate duplication of the genome is critical to successful cell division, the processes that occur during S phase are tightly regulated and widely conserved. Yeah, so I was remembering correctly, so that's good. So this G1 to S transition, this is basically the transition where a cell decides whether or not it wants to duplicate. So the iron depletion can lead to growth inhibition at this G1S transition, so it can stop the growth and presumably prevent cells from dividing. So these findings indicate other important physiological roles of the IRP-IRE signaling pathway beyond maintenance of iron homeostasis. So somehow iron is playing a role in cell duplication that is independent, or at least presumably independent, of its iron homeostasis. Anyways, linking IRP-IRE signaling pathways to human neurodegenerative disorders. So, however, recent findings indicate that iron accumulation-induced dysfunction of the IRP-IRE signaling pathway may contribute significantly to protein aggregation and neuron loss in Alzheimer's disease and Parkinson's disease. So, protein aggregation. So, frequently, proteins, if they get in high enough concentrations and if they are folded in particular ways or cleaved in other ways, they can actually precipitate at a solution. So these proteins are normally dissolved in the solution, right? So water is the universal solvent that is in your body that's keeping all these proteins solvated and afloat and not clumping together. But sometimes proteins will start, for lack of a better word, they start crystallizing at a solution, precipitating. And so this is also referred to as protein aggregation. And this protein aggregation can cause neuronal damage if it's dealing with some of the proteins that are found within the brain. So iron accumulation, recent findings indicate that iron accumulation-induced dysfunction of this particular pathway may contribute significantly to protein aggregation. So here they're not saying whether or not the iron itself is causing the problem, or how to phrase that. Is the iron dysregulation, well, here they are saying it. So I apologize. So the iron accumulation in the brain this is inducing dysfunction of this IRP-IRE signaling pathway, and this dysfunction of this pathway may contribute to the protein aggregation and neuronal loss. Now, granted, remember, this is a feedback loop, so if your IRP-IRE signaling pathway gets screwed up, that's also going to possibly lead to iron accumulation as well. So this might be one of the reasons why it's why we have progressive diseases, is because this IRP-IRE signaling pathway influences iron accumulation, and iron accumulation influences IRP-IRE signaling. Continuing, so this sheds new light on the pathogenesis and therapy of neurodegeneration observed in Alzheimer's disease and Parkinson's disease. So AD is the most common form of dementia in the elderly. The pathology of AD is characterized by synaptic defects. So the synapses are where your nerves meet to send signals from one nerve to another. So you can have defects in the synapses, and then you can have neuron loss and the formation of amyloid beta or A beta plaques in the hippocampus and cortical area. So just so you know, your brain frequently produces amyloid precursor protein, or APP, and then that amyloid precursor protein gets proteolytically cleaved into different peptides, or different smaller proteins. Now, certain enzymes will break it up into plaque-forming peptides, which are the amyloid beta, or these amyloid betas. And so these amyloid betas are a subset of the type of peptides that can be formed from the amyloid precursor protein. So some of the peptides are actually benign and don't cause problems, but some other ones that are, I think, 40 and 42 amino acids in length, those can form these plaques, and the formation of these plaques can cause neuron loss, right? So, and this is seen with Alzheimer's disease, and this happens in the hippocampus and the cortical areas. And the amyloid beta plaques contain amyloid beta aggregates and neurofibrillary tangles formed by aggregated forms of the microtubule associated protein Tau. So just know that the Tau is one of the proteins that associates with the microtubules, and I presume it is in high concentrations in the neurons. And because it's in high concentrations, that might be one of the reasons why it is associated with these neurofibrillary, or why it's associating with these AB aggregates. Let's continue. So amyloid beta is generated during protease cleavage of amyloid precursor protein, APP, and is critical in Alzheimer's disease pathogenesis. APP is a single transmembrane metalloprotein that can be cleaved through two main proteolytic procedures: amyloidogenic processing and non-amyloidogenic processing. The amyloidogenic processing of APP can generate amyloid beta peptides 40 to 42 amino acids in length, and among the amyloid beta peptides produced, amyloid beta 42 is more apt to form the fibrillar protein aggregate. So this amyloid beta 42 is the worst peptide to form these neurofibrillary, these fibrillar protein aggregates. So upregulation of APP expression and amyloid beta levels are associated with neuronal loss in Alzheimer's disease. Furthermore, increased levels of amyloid precursor protein can also be linked to amyloidosis in Down syndrome, since the APP gene is located on chromosome 21. So remember, chromosome 21, this is the chromosome that people with Down syndrome have an extra copy of. So they have 3 copies of it rather than 2. And so as a result, this amyloid precursor protein gene is actually found on chromosome 21. So people with Down syndrome are presumably might be producing a little bit more amyloid precursor protein than normal, and then this additional amyloid precursor protein might result in amyloidosis, which would be the just high levels of this. So rare disease that occurs when abnormal protein called amyloid builds up in your organs and interferes with their normal function. Amyloid isn't normally found in the body, but can be formed from several different types of protein. So Yeah, it might also be. Yeah, well, let's continue. Anyways, the increased levels of APP can be a pathological factor and a therapeutic target linked to amyloid beta amyloidosis and neuron loss in Alzheimer's disease and Down syndrome. So, however, recent findings suggest that increased amyloid precursor protein levels, aggregated amyloid beta deposition, and neuron loss in Alzheimer's disease can be induced by iron accumulation and disturbance of the IRP-IRE signaling pathway. So if you have too much iron accumulating, you can have basically Alzheimer's disease. So I need to look to see. Yeah, we'll keep on going. So a novel function of the iron response element is found in the 5' untranslated region of the amyloid precursor protein mRNA transcripts. So, and these are, it's in the 5' region, so it's earlier in the, it's before or in the promoter region of the mRNA. So it's in the early side or the left side of the mRNA. I might as well just get, so this is the example that we're talking about with the amyloid precursor protein, okay? So the IRE in the APP mRNA is located immediately upstream of an interleukin-1 response acute domain. So the interleukin-1 response acute domain, so remember interleukin-1, this is one of the cytokines that is just generally pro-inflammatory. So, okay, I didn't realize how big of a family it was. So there is this interleukin-1 responsive acute domain box, or acute box domain, that is right after the iron response element. So theoretically, both inflammation induced by interleukin-1 or that results in interleukin-1 can also modulate this activity. So is a group of 11 cytokines that play a central role in the regulation of the immune and inflammatory responses to infection or sterile insults. So yeah, we will come back to that possibly. So the IRE in the APP mRNA can bind specifically to the iron regulatory proteins, which can be abrogated by a mutation in the core IRE motif. In the presence of iron chelators, the translation of APP mRNA can be selectively downregulated. In contrast, iron influx or an increased iron level can enhance APP mRNA translation and amyloid beta generation. Therefore, these findings demonstrate that iron accumulation can be a pathological factor triggering the upregulated expression of the APP holoprotein and subsequent amyloid beta deposition. So if we have too much iron that is getting into the brain, it is possible that through this dysregulation process that you are having more amyloid precursor protein being synthesized, which can then result in more amyloid beta deposition. However, therapeutic strategies targeting the IRE in APP mRNA should alleviate amyloid beta deposition and neuron loss in Alzheimer's disease brains. The strategy of targeting the APP 5' untranslated region to reduce APP expression and amyloid beta formation has been validated by usage of novel IRE chemical inhibitors. So basically pharmaceuticals that can bind to this iron responsive element. And they do this to alleviate amyloid precursor protein and amyloid levels, as well as cognitive decline in this particular strand or this particular breed of an Alzheimer's disease mouse model. Identified potent IRE inhibitors such as Posifen. So let's look that up. Let's see if apparently there isn't a Wikipedia article on it, but so this is Posifen, just so that we are aware. So this is the structure that it has. Anyways, an identified potent IRE inhibitor such as Posifen, this These have been reported to be a bona fide amyloid precursor protein 5' untranslated region directed translational blocker that can reduce amyloid beta generation in the cerebrospinal fluid. So that's great. And this is in the cerebrospinal fluid of humans, and have entered clinical trials for AD therapy. So I wonder if they might still be in the trials. I would presume that the problem with an Alzheimer's disease drug is that the clinical trials probably take a very long time to be fully enacted because it is such a long haul disease. So anyways, let's keep on going. So in contrast, iron species can be a factor promoting reactive oxygen species generation and inducing amyloid beta aggregation. The increased iron levels in Alzheimer's disease patient brains is found to be relevant to the formation of neurofibrillary tangles. The therapeutic strategies using iron chelators plus IRE inhibitors may act collaboratively to alleviate amyloid beta aggregation and rescue neuron loss in Alzheimer's disease patient brains. Yeah, so one of the things that I want to mention is we want to have targeted. We don't want to just target all of the iron responsive elements in your brain, because that most likely wouldn't be benefiting you. So your brain has these iron responsive elements to help regulate iron, and so if you just throw a drug that indiscriminately screws up a lot of these iron responsive elements, then you might actually be making the brain more susceptible to iron dysregulation, which could theoretically cause more problems. So what I'm envisioning right now is there's sort of a catch-22. If your iron is becoming dysregulated, you're producing way too many amyloid precursor proteins, right? And so you're producing way too many APPs, which can result in these amyloid plaques forming. But on the other side, your brain is probably producing a lot of other proteins that are essential at helping regulate the high levels of iron in your brain. So if you just screw up all of those other proteins, you might result in more reactive oxygen species generation. So you might not be able to produce enough ferritin to help store that iron. You might not be able to produce enough transferrin. You might not be able to produce enough proteins that can help sequester the iron. Yeah, so you need to have targeted iron responsive element drugs or pharmaceuticals. So don't just hit all of them. Try to find the exact one that you want to hit. Anyways, we'll keep on going. So Parkinson's disease is the second most common neurodegenerative disorder, and the estimated incidence of Parkinson's disease is approximately 1% of the population over the age of 55. Parkinson's disease is characterized by progressive DA neuron loss. So dopaminergic neurons, these are neurons that are associated with dopamine in some way. So you have the progressive dopaminergic neuron loss in the SN as well as the Lewy body formation in the affected brain areas. So I do not remember what SN is. So SN brain. Oh, the substantia nigra. Okay, so yeah, it's just one of the parts of the brain. And it's one of the parts in the brain that produces neuromelanin, if I'm remembering correctly. So regardless, you have this dopaminergic neuron loss in these areas. Anyways, the pathogenesis of the progressive dopaminergic neuron degeneration in Parkinson's disease is still unclear as of yet, and no effective therapy has been developed to alleviate the progressive dopaminergic neuron degeneration in Parkinson's disease. However, recent new findings on the roles of IRP-RE signaling pathways in Parkinson's disease add to the pathogenesis and therapy for iron iron-relevant DA neuron degeneration in Parkinson's disease. So the involvement of IRP in DA neuron degeneration can be supported by 2 in vivo studies on IRP2 knockout mice models. So remember, you have 2 iron regulatory proteins, and there is a knockout with the iron regulatory protein number 2 in this mouse model. And the involvement of these proteins in the dopaminergic neuron degeneration can be supported by 2 studies on these knockout mice. Wait, did I? Let's see, 57. I'm trying to remember if I've gone over this paper before. No, not this one. Okay, continuing. So, in the middle to late age, 18 to 24 months, mice lacking the IRP2 will develop abnormal motoric Parkinson's disease-like behaviors, including tremors at rest, abnormal gait, and bradykinesia, which I always need to remind myself, nope, I do not want to print, I want to search. So it means slowness of movement and is one of the cardinal symptoms of Parkinson's disease. So if you move slowly, that would be the bradykinesia. So furthermore, significant iron accumulation in the brains of the IRP2 knockout mice precede the onset of dopaminergic neuron degeneration and Parkinson's disease-like symptoms by many months. So you basically have iron accumulation in the brains of these iron regulatory protein 2 knockout mice, and then that precedes, or that comes before, these Parkinson's-like symptoms. So that's interesting. So what this is indicating to me is the iron, we're dealing with the chicken or the egg scenario. Is the iron accumulating because you have dysregulation of the iron regulatory proteins, and then that causes the Parkinson's, or is that causing the neurodegeneration? Or is it the iron accumulates first, and then you have the dysregulation of the IRP-IRE system, and that leads to Parkinson's? It's a feedback loop, right? So it's probably both. Let's continue. Anyways, you have this, and this is all accompanied by the deposition of ubiquitin-positive protein aggregates and inclusions in the mouse brain. So we're finding these protein aggregates, and they're ubiquitin positive. So that means they've been ubiquitinated, and they are supposed. If a protein is ubiquitinated, that can be used as a way to signal to the cell where that protein needs to be put into. Frequently, it is for targeting to take it to the proteasome, where it gets degraded and broken down into amino acids. So here, when we're dealing with a ubiquitin positive protein aggregate, this to me sounds like a protein that was targeted and was trying to be sent off to be destroyed by the proteasome, but ended up not being destroyed by the proteasome for some reason. That's not necessarily the case, but that's what it seems like to me. There are other things that ubiquitin is used for, so I'll just leave that as an open-ended thing. Anyways, the dopaminergic level the dopamine level in the dorsal striatum of the IRP2 knockout mice decreased approximately 20%, which is significantly compared with that of control mice. This is suggesting that IRP2 knockout induced dopaminergic neuron degeneration in the mouse brain. Previous findings demonstrated that iron retention and iron-dependent toxicity to dopaminergic neurons can be induced by nitric oxide through facilitation of interactions between IRP and IRE to inhibit translation of ferritin, ferroportin, and other iron metabolism relevant proteins. Furthermore, NO, nitric oxide, can inhibit amyloid precursor protein translation to aggravate the iron retention in dopaminergic neurons. Wait. So previous So if you have nitric oxide, this will facilitate the interaction with the IRP and the IREs. So nitric oxide can inhibit the translation of ferritin and ferroportin and other iron metabolism relevant proteins. So that's very interesting that nitric oxide can have that effect. So furthermore, nitric oxide can inhibit APP translation to aggravate the iron retention in dopaminergic neurons, as APP stabilizes ferroportin to promote iron export. Oh, so this makes sense. So why does your body produce more amyloid precursor protein in the first place? Well, the amyloid precursor protein, one of its jobs is to stabilize the ferroportin, which helps pump the iron out of the cells. So if you could imagine, if you have too much iron in your cell, you're going to produce a lot of this amyloid precursor protein to try to get it out of the cell if possible. So nitric oxide is supposed to be a causative factor upstream of negral iron accumulation and dopaminergic neuron degeneration in Parkinson's disease pathogenesis. These findings suggest a close association of iron and the iron regulatory protein IRE signaling pathway with dopaminergic neurodegeneration in Parkinson's disease. Fascinating. Anyways, furthermore, previous evidence suggests that iron accumulation and dysfunction of the IRP-IRE signaling pathway can be linked to alpha SYN-induced toxicity of dopaminergic neurons. So alpha-syn is a central protein involved in the pathogenesis and therapy of Parkinson's disease. So Lewy bodies consisting of aggregated alpha-syn can be found, and I believe alpha-syn is named after senility. One second. Yeah, so alpha-synuclein. This is a protein that is encoded by the SNCA gene, and it's abundant in the brain, while smaller amounts are found in the heart muscle and other tissues. And in the brain, alpha-synuclein is found mainly at the tips of neurons in specialized structures called presynaptic terminals. Within these structures, alpha-synuclein interacts with phospholipids and proteins, and presynaptic terminals release chemical messengers called neurotransmitters from compartments known as synaptic vesicles. The release of neurotransmitters relays signals between neurons and is critical for normal brain function. So function of this is not well understood, so just so you know, this is the protein that we are dealing with. So this can be linked to, if you have too much of it, you can have alpha-synescin-induced toxicity of dopaminergic neurons. Alpha-synescin is the central protein involved in the pathogenesis and therapy of Parkinson's disease. So Lewy bodies consisting of aggregated alpha-synescin can be found in almost all Parkinson's disease patient brains. Alpha-synescin mutations are known to be linked to onset of the familial form of Parkinson's disease, or FPD. Furthermore, the increased levels of alpha-synescin protein in the brain due to gene duplication or triplication is also linked to the onset of familial form of Parkinson's disease. So if you have too much of this alpha-synescin gene, too many of these alpha-synescin genes in your body, you might be producing way too much of it. And if you're producing way too much of a protein, the concentration of that protein can be high enough so that it starts to crystallize or fall out of solution or form aggregates. However, human post-mortem brain studies have demonstrated that while the mRNA levels of alpha-synescin in Parkinson's PD brains are either unchanged or decreased, higher levels of the insoluble alpha-synescin protein occur during the Parkinson's disease progression. So what does this tell you? This tells you if the mRNA levels are the same, that means that it is somehow being upregulated at the mRNA level. And this has to do with the translation. So the translation of the protein is what's being regulated. So it's not the transcription of the DNA into mRNA. It's not that where it's being regulated, because that seems to be the same in the Parkinson's versus non-Parkinson's patients. Now, how do I phrase this? And it doesn't have to do with the degradation of mRNA. So you know how sometimes uh, the mRNAs can be processed by the endonucleases that can alter the levels of mRNA in the cell. Well, um, apparently there is not a difference, not a statistically significant difference in the Parkinson's disease patients and the normal deaths, right? So what's going on is probably something to do with the translation in the five or with the iron regulatory proteins or some regulation that's happening in the promoter region of the mRNAs rather than endonuclease activity or the transcription. So let's continue. So the in contrast, disrupted iron metabolism in the SNs, the substantia nigra, is a common feature of Parkinson's disease, and studies have shown that iron levels can be upregulated to almost twofold uh, in a single substantia nigra dopaminergic neuron in the Parkinson's disease patient brains. The elevated iron levels in PD patient brains are thought to be linked to the accelerated deleterious formation of alpha-synuclein protein aggregates. So furthermore, novel evidence has shown that elevated iron levels in the brain can cause enhanced alpha-synuclein protein translation in the brain. A unique RNA stem loop with the IRE motif, this is found in the 5' untranslated region of alpha-synuclein mRNA. The IRE stem loop of the alpha-synuclein transcript spans 2 exons. IRPs can bind with the IRE of alpha-synuclein mRNA and inhibit translation of the alpha-synuclein transcript. However, iron can reverse the translation inhibition by alpha-synuclein by IRP and therefore facilitate alpha-synuclein expression and aggregation. So if you have a lot of iron, you can kick off. So that's basically what's happening here is normally the iron responsive iron regulatory proteins are preventing synuclein from being synthesized in too high concentrations, but if you have way too much iron, you are kicking off more of these iron regulatory proteins and as a result producing more of this alpha-synuclein. So translation of alpha-synuclein mRNA can be inhibited by new IRE chemical inhibitors, and these findings suggest a pathological relevance of iron in the IRP-IRE signaling pathway of alpha-synuclein-dependent toxicity of dopaminergic neurons in Parkinson's disease. Iron chelators and new IRE inhibitors should alleviate iron-dependent alpha-synuclein toxicity and dopaminergic neuron degeneration in Parkinson's disease. The detailed IRE sequences and stem loop structures, as well as maps of the IRE stem loop and alpha-synuclein and APP transcripts are illustrated in figure 2. So here are examples of the iron responsive elements of different mRNAs for encoding for different proteins. So here we have the H-ferritin, and here we have the L-ferritin. Here we have amyloid precursor protein, and here we have alpha-synuclein. And frequently we have this CAGU, 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 CAGU. We frequently have this appear. Oh, and this one's CAGA. So this one's a little unique. But regardless, this little loop at the end of the stems, this is frequently conserved in a lot of different iron regulatory element proteins or iron regulatory elements that are encoding for different proteins that need to be up or down regulated in response to iron presence. Now, and I should say these are all 5' untranslated regions. So these are all the ones that will be upregulated in response to iron. So if you want, you can look at this yourself, but you can pause it and look, but I'm not going to focus too much on this right now. So the iron and IRP-IRE signaling pathway also seems to be implicated in the pathogenesis of alpha-Lou Gehrig's or ALS, which is amyotrophic lateral sclerosis. I think it has another Lou Gehrig's, yeah, Lou Gehrig's disease. Okay, so ALS, so amyotrophic lateral sclerosis, or Lou Gehrig's disease, and this is extremely debilitating. It's very disheartening to see this happen to people. But the iron and the IRPRE signaling pathway also seems to be implicated in the pathogenesis of Lou Gehrig's disease. So ALS is a progressive neurodegenerative disorder induced by multiple pathogenic factors and is characterized by the progressive and selective loss of motor neurons in the cerebral cortex, brainstem, and spinal cord. So alpha-synuclein positive inclusion bodies can be identified with ALS. Increasing evidence indicates a pathological role of iron dysregulation in neuronal cell death in ALS. The role of iron in the pathogenesis of ALS is supported by the therapeutic effects of iron chelation strategy in ALS mouse models. Furthermore, a potential IRE has been computer predicted to exist in the 5' untranslated region of the mRNA encoding for C90RF72, which is a key ALS-linked gene. So this is a gene that people with ALS, they have a mutation in it, or it's a little different from normal people, and this is one of the genetic factors that could possibly influence Lou Gehrig's disease formation. And this also happens to have a computer-predicted mRNA, a computer-predicted iron response element. So a computer can look at the mRNAs and they can see if there are large areas of pairing, and these large areas of pairing can determine whether or not you have secondary structure in a certain location of the mRNA. And you can, if you have this stem and if you have a loop, and if the loop has some of these conserved nucleotides, then this could indicate that this might be an iron regulatory or an iron response element, or something that is like an iron response element. Anyways, this RNA structure is proposed to post-transcriptionally regulate the expression of the C90RF72 protein in response to iron dyshomeostasis. So let's look this up real quick just to... Chromosome 9 open reading frame 72 is a protein which in humans is encoded by this gene. Excuse me. So yeah, there are mutations in this that are associated with the FTD and ALS. So yeah, there is a mutation in some people in this particular gene that is associated with getting ALS. Anyways, this RNA structure is proposed to post-transcriptionally regulate the expression of C9ORF72 protein in response to iron dyshomeostasis. Such a hypothesis can be supported by previous findings on the increased levels of iron and ferritin in patients with ALS. Thus, a potential mechanistic link may be present between iron and the iron modulation translation of C9ORF72, which is associated with ALS pathogenesis. So although none of the point mutations in IRP1 and or IRP2 have been confirmed to cause human diseases, you know what? I think I'll stop here because we're at an hour, and this is a good shift going from neurological diseases to other diseases. So with that, I'm going to head on out. But the notes that I want to make is that it is very possible that this iron dysregulation is either the accumulation of iron is causing the iron dysregulation, or the iron dysregulation is causing the accumulation of iron. Either way, we are getting a buildup of iron in the brain. And this buildup of iron is then going to screw up with the translation of a lot of the mRNAs that are within your brain. Okay, so what this would indicate to me is that the brain is now going to be producing way too much of these proteins, and as a result of producing too much of these proteins, you can start getting more aggregates of these proteins. So remember, these proteins are there to protect you. These proteins are there to deal with the iron that's in your brain, right? But if basically you can saturate that process to the point where the proteins are no longer benefiting you, but they're actually hurting you. So with that, we can run we can run into a serious situation where no matter what you do, your brain's not able to heal itself. So as a result, we need to look to pharmaceutical methods of sequestering the iron. That might be one of the important things that can be done in order to help prevent this from happening. So some people might argue that maybe the reduction of iron in someone's diet should be considered. Uh, Perhaps this might be one of the reasons why people who drink a lot of coffee are associated with less neurodegenerative diseases, because coffee is also able to inhibit iron absorption, right? So coffee I don't remember the exact mechanism of action, but coffee and teas are able to prevent excessive absorption of iron. And so preventing this excessive absorption can be extremely beneficial, or might be extremely beneficial for some people in preventing neurodegeneration. Interesting. So, as I drink coffee right now, um, so yeah, that's all very important. Now, so in the context of the current pandemic, I do believe that many of these diseases, especially the diseases that are able to infect neurons, they might play an extremely powerful role in the dysregulation of the iron homeostasis in the neurons. And also, I would probably also argue that these coronaviruses explicitly are doing a very good job at causing iron dysregulation in the rest of the body to help liberate the labile iron in order to be used for the viral replication. So how to phrase that? This event might be able to dysregulate iron not only in the body but also dysregulate it in the brain, which can then result in possible neurodegenerative complications down the road. And this is probably why frequently we find the RNA for coronaviruses in the cerebrospinal fluid of people with these neurodegenerative diseases. So perhaps those other coronaviruses just aren't as good at doing it, and therefore, like, they're not as good at killing people, right? So as a result, we're mostly fine, but it appears as though the more serious coronaviruses might seriously cause this dysregulation and cause more neurodegenerative disorders. And I cannot pronounce that word for the life of me. So with that, I'm going to head on out. Thanks for watching, and hopefully I will see you later today.